This video is on Carnivore Protoparvovirus 1 in dogs, otherwise known as canine parvovirus. To be discussed are methods of sample collection, preservation, and transportation, as well as laboratory tests used for diagnosis. The characteristics of this virus concerning its growth, infection, and spread will also be discussed.
In order to test for Carnivore Protoparvovirus 1 in dogs, you would take fecal samples to send to a laboratory, since large amounts of virus particles are shed in the feces of infected animals. Fecal samples need to be preserved and transported to laboratories. The sample should be approximately 5 grams. The sample should be fresh, no more than 6 hours old, or, if refrigerated, 24 hours old. They should be placed in a plastic container or 2 plastic bags, effectively being double bagged. Morphology of the virus. The three-dimensional structure is determined using X-ray crystallography. The morphology is characterized by small isometric particles containing a single-stranded DNA genome. Growing the virus in cell cultures. The virus is unable to grow in standard cell cultures. The best way to grow the virus in a laboratory is using the stable feline kidney cell line FE. The following laboratory tests are used to confirm viral infection. Antibodies can be detected in most cases after 3 days of symptoms using an enzyme-linked immunosorbent assay or radioimmunoassay. IgM antibody assay detects recent infection, and a nucleic acid hybridation can be used to test for the presence of viral DNA.
When making our diagnosis, same symptoms such as diarrhea and vomiting can be caused by different pathogens. Examples of bacteria would be Clostridium perfringens, E. coli, and Salmonella, and viral diseases like distemper. It is therefore very important to be able to find the right pathogen and exclude the others. Bacteria are quite easy to differentiate, especially from a morphological point of view, since bacteria are ultimately larger in size. Additionally, note that parvoviruses are among the smallest DNA animal viruses, and they also replicate differently. This can be observed in cultures with different stains and under a light microscope. More complex tests can, of course, be made, such as PCRs and ELISA tests, but wouldn't necessarily be needed to rule bacteria out. It gets a little trickier when differentiating with other canine viruses, but the laboratory tests discussed in the previous slides can all serve this purpose. This disease needs early and fast diagnosis since it is highly contagious. We will therefore not discuss how to differentiate with identification through electron microscope and virus isolation, which can take quite long. Other tests, such as latex agglutination and hemagglutination, usually lack reliability and specificity. The enzyme-linked immunosorbent assay, also known as ELISA, is one of the most practical methods for rapid diagnosis of antibody detection in feces or blood samples with immunoglobulin G and immunoglobulin M antibodies. The polymerase chain reaction, PCR analysis, is widely applied for laboratory diagnosis because of its high sensitivity and specificity. It can rapidly detect gene sequences specific to Carnivore Protoparvovirus 1, notably sequences of NS1 and NS2, which are non-structural proteins. Note, however, that without specialized equipment, this test isn't as accessible for field veterinary clinics. A new technique, the loop-mediated isothermal amplification, also known as LAMP, is very fast has an extremely high sensitivity and produces an end product visible to the naked eye. It also doesn't rely on thermal cycler, and so this is a bit more accessible. All of these are specific to either antibodies or a gene sequence of the canine parvovirus and can therefore confirm and rule out other viruses. We can also confirm canine parvovirus once the puppy has died and a biopsy is made. Lesions in the GI tissues are clear and characteristic of the pathogen. A very important part of the replication cycle of a virus is the eclipse phase. It is the period of time between the penetration of the virus into the host cell and the production of the first new virus particle. This is very critical, as any disruption to the phase inhibits the release of high numbers of virus particles. In general terms, virions, shown here by the red arrow, must bind to a specific receptor and then cross the plasma membrane barrier by a clathrin-mediated endocytosis to deliver its genome to an appropriate site for uncoding and decoding. For our non-enveloped CPV, canine parvovirus, which needs to replicate in the nucleus and lack fusion proteins, the same pathway is used, but the uncoding process is still poorly understood. Studies agree that the partially uncoded virions are transported via microtubular transport to the nucleus, where the final uncoding step occurs, and then replication takes place. Canine parvovirus is a relatively new virus. Research suggests it emerged approximately 60 years ago. And like most fast-replicating entities, it is continuously evolving and prone to mutations. This gives rise to new antigenic types and virus mutants within dog populations. Here we can see that although origins of the virus are debatable, the original CPV type 2 went extinct and is now replaced by CPV2A and 2B. And here we can see the relevant amino acid differences between the virus types. Note that CPV2B has since largely replaced CPV2A, which shows the rapid mutation rate of this virus. How, where, and why these mutations occur are discussed in many studies. Some of the known mutants, and most successful ones, are selected by improved binding capacity to the canine transferrin receptor. Without going into too much details, this is the receptor to which the virus binds to infect a host cell. So if it is more efficient in doing so, the virus has better chances in infecting and replicating. They can also be very successful by extending the range of viable hosts. Indeed, with the newer antigenic types of the virus CPV2, as discussed previously, can include both dogs and cats as potential cell hosts. There are also antigenic mutations with different potential to bind to certain monoclonal antibodies. And then, of course, their reactivity to viral neutralization tests can differ, so we have more resistant types. All these more or less minute mutations and different infecting virus types influence the contagion rate and the susceptibility of the young animals to be infected.
Transmission of the virus. The virus is highly contagious. It can spread through direct contact with an infected animal or indirect contact with regards to contaminated objects. It spreads using the fecal-oral route or by oronasal exposure to contaminated fomites. It is predominantly shed in feces, but it may also be found in vomit, urine, and saliva of infected animals. It is environmentally resistant and can survive for a long time outside the host. This contributes to the reason why it is so contagious. The virus is an intracellular pathogen and it evades the immune responses by acquisition of mutations in their capsid proteins. The viral capsid is formed by structural proteins VP1, VP2, and VP3. The structural protein VP3 is formed by the removal of part of the amino terminus of VP2. It is a proteolytic product. This N-terminus contains a neutralizing site conserved in different viral isolates, which in the form of synthetic peptides is able to induce neutralizing antibodies and protection in the host cell.